I was going there's what's this place. I wanted to post our next time we have 2 more on topic.
All right, I'd like to welcome everybody to our Biological Chemistry Faculty Research Mini Symposium. We have 2 excellent faculty candidates speaking today, Whiteley and Scott Coyle. I'm going to go ahead and introduce Aaron now. Aaron did his PhD with Daniel Courtney at UC Berkeley, where he worked on cyclic di-AMP signaling in Listeria, and also how Listeria sense changes in the post-cytosolic environment, including changes in redox state. Then he moved east to Harvard Medical School, where he did a postdoc being co-mentored by John Mechalanos and Philip Crenches, where he worked on the biochemistry of nucleotidal and their role in bacterial pathogenesis and also in eukaryotic immune cell signaling. His talk today is titled Bacterial C-gas-like Enzymes at the Host-Pathogen Interface.
Well, thank you so much for that introduction, and thank you all for coming. I'll go ahead and just try and project, but tell me if you can't hear me. So today I'm going to be talking about these bacterial C-gas-like enzymes. But before we get into that, I want to give you a feel for the kinds of questions that interest me as a researcher. So I'm interested in the interaction of bacteria and their eukaryotic hosts. And although that term eukaryotic host sort of implies that this is just bacterial antagonism of eukaryotes, I actually think it's much more broad than that. And you can imagine many different relationships between bacteria and eukaryotes that can be both mutualistic, commensal, or antagonistic, such as the case of bacterial pathogens. Specifically, I'm interested in a family of chemicals that are exchanged between bacteria and eukaryotic hosts that are called pathogen-associated molecular patterns, or PAMPs. These are molecules that are often fundamental to bacterial physiology, and they're so core to the bacteria that eukaryotes have evolved a set of germline-encoded receptors that specifically recognize these core chemical components. The signaling that is elicited by pathogen-associated molecular patterns, or PAMPs for short, from eukaryotes is called innate immune signaling. And that term innate is to refer to the fact that these are germline-encoded receptors that are often found across all vertebrates. The reason that I'm interested in understanding PAMP signaling, innate immune signaling, and this interaction is because it's fundamental to health and human disease. And there's many different disease conditions that probably everyone knows of that are dominated by how bacteria interact with eukaryotic hosts, and at least a portion of those are determined by these chemical signals that are exchanged. Today, by way of an outline, I'm going to tell you about a family of proteins called bacterial C-gas-like enzymes. These are structural homologs of a eukaryotic protein called C-gas that we'll get into in a moment. And what's interesting about these proteins is that they synthesize a family of nucleotide second messengers that hadn't previously been identified. Those nucleotide second messengers participate in innate immune signaling because they're detected as PAMPs. So the chemicals that I told you about before that are involved, we think, in core bacterial processes, although we don't yet understand what those are. The long-term goal of my independent research is to understand how these novel nucleotide second messengers are participating in health and human disease. So with that, I'll just get into the more specific background. So C-gas, which is in the title and is going to be pretty important for today, is an enzyme that's encoded by vertebrates, but we know the most about the mammalian form of the enzyme. It's an enzyme that resides in the cytosol of cells where it acts as a physical sensor of double-stranded DNA that's become mislocalized in that compartment. Upon interaction with double-stranded DNA, C-gas becomes activated to synthesize this molecule that I'm showing you here, cyclic GMP-AMP, or C-GAMP. C-GAMP is made from the cyclization of ATP and GTP into this molecule, where it then acts as a nucleotide second messenger that can diffuse throughout the cytosol, bind the protein receptor sting, and elicit type 1 interferon. And what I want you to appreciate about this pathway is that it links sensing of double-stranded DNA that's become mislocalized in a different compartment with an antimicrobial response. So this is, you can imagine a DNA virus invading into the cytosol, or a bacterium that through lysis has released DNA. And although that this pathway has principally been understood through the lens of antimicrobial immunity, what's become increasingly appreciated and what's emerging in the field is that this pathway is also very important for eliciting immune responses to cancer. And in fact, CGAMP-like molecules are being used in the clinic right now as potential adjuvants to cancer immunotherapy. But the reason that I became interested in this pathway is actually because it fits into a broader understanding of a family of PAMPs that are made by bacteria. So in addition to CGAMP, STING, that receptor that turns on type 1 interferon, is also capable of binding and signaling from the molecules I'm showing you here, cyclic dinucleotides that are made by bacteria and have been studied for over 30 years. So cyclic di-GMP, cyclic di-AMP, and a bacterial form of the CGAMP molecule are all cyclic dipurines that are capable of eliciting type 1 interferon. And so STING has this dual role in seeing both bacterial-derived PAMPs and seeing this host molecule that's downstream of bacterial PAMPs. The bacterial form of CGAMP is synthesized by the enzyme I'm showing you here, DNCV. So this was discovered in Vibrio cholera in 2012 in the Mecolanos lab, and what was found was that DNCV, this synthase, produces the molecule I'm showing you here, and that's important for motility and virulence in Vibrio cholera. Interestingly, DNCV and CGAS both synthesize very similar molecules, although they're not identical. And so I'm just highlighting that the difference between these two molecules is the linkage here, either 3', 5' between the two nucleotides for DNCV, or 2', 5' for the CGAS-derived molecule. So they're subtly different, but the shared ability of these two enzymes to synthesize such similar molecules comes from the fact that they're actually structural homologs of one another. So this wasn't initially appreciated because there's no discernible amino acid identity shared between DNCV and CGAS, but when the crystal structures of these two enzymes were solved, what was found is they're both pol beta-like nucleotidyl transferases. So this is a polymerase-like enzyme fold, and you can even superimpose the two structures on one another in order to appreciate the similarity. So here in green is DNCV, and in magenta is CGAS. I was transfixed by this relationship between these two enzymes when I started my postdoc, because DNCV is in fact a bacterial CGAS-like enzyme, and the shared ability of these two protein folds to make molecules that have this innate immunostimulatory role suggested to me that perhaps if we could find additional bacterial CGAS-like enzymes other than DNCV, we would continue to expand the repertoire of adjuvant-like molecules that are being made by bacteria. And just to take that one step forward, I think it's important to identify these other bacterial CGAS-like enzymes, mostly to understand, getting back to that interaction of bacteria with their eukaryotic hosts. And so I set about trying to design a way in order to find additional bacterial CGAS-like enzymes like DNCV. And to start, I took what I like to call an unsophisticated approach. And that really started by looking at the operon organization of the DNCV gene in Vibrio cholera. So here I'm showing you that Vibrio cholera encodes DNCV adjacent to a gene which encodes the receptor for 3', 3' CGAM. And what I found was that that operon, that DNCV operon, could also be found in other bacteria. Within those bacteria, and here I'm showing you an E. coli, and notably this is not the K12 E. coli that you work with in your lab conventionally, it's a different strain of E. coli, but this strain encoded a DNCV-like operon. E. coli DNCV also is capable of synthesizing CGAM, and that's because these two enzymes are 60% identical. But just upstream of the E. coli DNCV operon was what looked like a gene duplication event. So here we found genes that were highly similar to the 3', 3' CGAMP receptor, and another gene that we named CDNE that looked weakly to be perhaps a polymerase-like enzyme, but shared no detectable amino acid identity to DNCV. So it appeared to perhaps fit into this mold of a bacterial CGAS-like enzyme, and I set about asking whether CDNE is also capable of synthesizing CGAMP, and if not, what else is it doing? To ask that question, I took a biochemical approach. And so here, We're incubating, the format of this assay